Okay, so we're going to be talking about zoonotic diseases, and basically like how they're treated and also how they can be prevented.
So let's start with a brief overview of zoonotic diseases. So what are they? Well, they're diseases spread from animals to humans. Every year, over 10,000 Americans are infected with zoonotic diseases. 6 out of 10 infectious diseases in humans are zoonotic diseases, and to find cures and vaccines to zoonotic diseases takes groups of disease detectives, laboratorians, physicians, and veterinarians. So can anyone tell me what the difference is between a virus and a disease?
So it's definitely okay if you guys don't know what a virus and disease is. You can feel free to type in like any guesses if you want. But for me, I was definitely confused about it at the beginning. So a virus is basically something that causes a disease. So a disease would be something like a cough or like a runny nose. And a virus is basically what causes those symptoms. And so the two terms essentially describe the same thing. So that's why people use them interchangeably. So yeah, just keep that in mind during the presentation. Um, I think we also use virus and disease a lot. So I think it's good to know the difference like ahead of time.
So, I'm sure you're all familiar with COVID-19. You know, it's a deadly virus that has affected the entire population. People have been forced to go into quarantine, lost jobs that they needed, and have even lost their lives. When we go out now, a new part of our essentials are a mask, and we're not allowed in the majority of areas without one. But what is COVID-19 really? What do you think it is?
Yeah, so you guys can feel free to type in the chat what you think COVID-19 is and how it's affected you. Yes, it's definitely a virus and it's made us stay inside so we can't go out. Yes, a cough is a symptom of it. Yeah, and a lot of people have been affected by it, including a lot of our loved ones and people that we know. Okay, I think it would be good to continue with it, right, Sydney?
An overview of COVID-19, background and origin. Coronavirus disease is an infectious disease caused by a newly discovered coronavirus, which is any group of RNA viruses that cause a variety of respiratory, gastrointestinal, and neurological diseases in humans and other animals. The virus originated in Wuhan, China, and experts think that the virus started through an animal sold through an animal market. The most common symptoms of this virus include fever, dry cough, and tiredness. COVID-19 has impacted people worldwide. It has slowed down the economy through a decrease in retail shopping, restaurants, and more. People aren't able to travel, and when they can, they may be subjected to quarantine or a check once arriving. Overall, daily life has been impacted in so many ways involving quarantine and a decrease in social activities. Schools and workplaces have closed or shut down, meaning that people are losing their jobs while their children are staying home 24/7. People all around the world have been negatively affected due to this pandemic. So, how can we prevent COVID-19? Don't go out as often, always wear a mask when you leave the house, and wash your hands frequently. Stay safe.
I'll be talking about the current vaccines and methods being used to treat COVID-19, as well as the process scientists have to go through to develop the vaccine. As you may know, scientists have been working hard trying to find an effective vaccine for COVID-19. There are 3 essential phases a vaccine has to go through before being fully approved. Preclinical testing does not count as a phase, but it is when scientists test a new vaccine on cells and then give it to animals to see if it produces an immune response. There are 87 confirmed preclinical vaccines in active development. In phase 1, scientists give the vaccine to a small number of people to test the safety and dosage as well as to confirm that it stimulates the immune system. In phase 2, scientists give the vaccine to hundreds of people split into groups, such as children and the elderly, to see if the vaccine acts differently in them. These trials further test the vaccine's safety and ability to stimulate the immune system. In phase 3, scientists give the vaccine to thousands of people and wait to see how many people become affected, compared with volunteers who received a placebo. Phase 3 trials are large enough to reveal evidence of relatively rare side effects that might be amiss in earlier studies. The limited approval stage allows for a limited amount of people to receive the vaccine. To actually become approved, regulators in each country review the trial results and decide whether to approve the vaccine or not. During a pandemic, like COVID-19, a vaccine may receive emergency use authorization before getting formal approval. Once the vaccine is licensed, researchers continue to monitor people who receive it to make sure it's safe and effective. In times of need, scientists can accelerate vaccine development by combining phases. Some coronavirus vaccines are now in phase 1/2 trials. For example, in which they are tested for the first time on hundreds of people. There are currently 4 vaccines that are approved for limited use and 2 approved for early use. However, these vaccines are all from outside the US, and I would rather focus on the ones being produced within our country, as they have been discussed far more in the news. Pfizer has also recently requested for emergency use authorization for their vaccine, as they have proven that their vaccine is 90% effective based on early data from its trials. Moderna has also stated that they are close to seeing early data from their phase 3 clinical trial. The vaccines from Pfizer and Moderna are mRNA vaccines. Like other vaccines, mRNA vaccines work by training the immune system to recognize a threat like a virus and begin producing antibodies to protect itself. But while traditional vaccines often use inactivated doses of the organisms that cause the disease, mRNA vaccines are designed to make the body produce those proteins itself. Messenger RNA, a molecule that contains instructions for cells to make DNA, is injected into cells. In the case of COVID-19, mRNA vaccines provide instructions for cells to start producing the spike of protein of the new coronavirus, the protein that helps the virus get into the cells. On its own, the spike protein isn't harmful, but it triggers the immune system to begin a defensive response. As Bill Gates, who has supported companies like Moderna and BioNTech through the Gates Foundation, has described it, you essentially turn your body into its own manufacturing unit.
Zoonotic diseases are very infectious and caused by a pathogen, which is an infectious agent such as a bacterium or a virus that has transferred from an animal to a human. It's important to protect ourselves from zoonotic diseases by eliminating the pathogen from the animal. There are many things we can do to help prevent these diseases. First, the most important thing is personal hygiene. It's critical to wash your hands both before and after handling animals. If you don't have access to soap and water, use an alcohol-based hand sanitizer. Another important aspect is the hygiene of the animals. Those who take care of animals and pets should be checking on the health status of the animals often, and then isolating the sick animals and reporting the dead ones. Be cautious of not spreading possible pathogens when cleaning areas around ill animals. Environmental maintenance is equally important, and you should make sure that there is proper ventilation to protect both the animals and workers, and keep animal housing areas clean, safe, and organized. If needed, take vaccinations against certain diseases. Finally, make sure to avoid bites and scratches from animals, as those can quickly spread infections and diseases. 60% of diseases in the world are zoonotic, so they spread more often than you would think. Zoonotic diseases are responsible for at least 2.4 billion cases of human illness and about 2.2 million deaths per year, which is really significant. It's super important we all do as much as we can to help prevent them from occurring and spreading.
Okay, influenza, aka the flu. The one you get shot in the arm for, and there's also the one where you breathe it. But the one that goes through your bloodstream is just influenza is a contagious acute respiratory illness caused by influenza viruses and is an infectious / zoonotic disease. When it comes to how flu spreads, most experts believe that the virus spreads mainly by tiny droplets made when people with flu cough, sneeze, or talk. These droplets infect each other when in contact with their mouths or noses, which means that if you just cough on a surface and you touch that and then touch your nose or face, then you would be infecting yourself with that. In the US, on average, about 8% of the population gets infected each season, with range between 3 to 11%, depending on the season. Wow. People with a high risk from the virus include people 65 years and older, people of any age with certain chronic medical conditions, pregnant women, and children younger than 5 years. Methods like covering coughs, frequent hand washing, and staying away from people who are sick help slow the spread of germs, which help slow the spread of the virus. There is also a very important method of flu prevention called getting a yearly vaccine.
So now we're going to talk about vaccines. So they're basically substances which are injected and it protects us from deadly diseases like the flu and COVID-19 like Benjamin and Sydney talked about. So there is a weakened or deactivated version of the microbe in the vaccine. And when the person gets vaccinated, they develop immunity against the disease. There's a lot of debate revolving around whether vaccines are safe, and the truth is yes, they are safe. All vaccines are tested by the FDA and approved before it can be given to the public. So like you guys saw when Kathleen talked about it, there's many stages to make sure that it's safe for everyone to take it. So now let's talk about the different types of vaccines. There are several different types of vaccines, and we're going to learn about 3 of them. So the first one is live attenuated vaccines. So they use the weakened form of the germ which causes the disease. And since they are very similar to the disease, which has to be protected, they create a strong and long-lasting immunity. So just 1 or 2 doses could result in lifelong immunity. But since they do contain a small amount of the germ, even though it's weakened, it may not be safe for people with weak immune systems and people who have long-term health problems. And so they also need to stay chilled and they don't travel well. And mumps, measles, rubella, rotavirus, smallpox, chickenpox, and yellow fever are diseases prevented using this kind of vaccine. And the next one is inactivated vaccines. They use the killed version of the germ, but they're usually not as strong as live vaccines. So they require booster shots or several doses in order to gain immunity. The next one is subunit recombinant polysaccharide and conjugate vaccines. They use specific pieces of the germ, like its protein, sugar, or capsid, and they provide very strong immunity as the target key parts of the germ. And they can also be used on almost everyone who needs it, even people who have weak immune systems. And the only limitation is that the booster shots may be required, and shingles, hepatitis B, and whooping cough are some diseases which are prevented using this kind of vaccine.
Okay, so In summary, a mutation is a change in something, but not entirely. It can either be an addition to the original thing or a complete modification or change to it. In this case, we'll be discussing about the mutations within the DNA and in viruses such as influenza, or more commonly known as the flu. These two mutations are the most basic examples that occur within the body. Though both are related to one another, there are a few key differences between them. When a mutation occurs in the DNA, the cell or cells become harmed and will change accordingly. Some outcomes include sickle cell anemia, shown on the image at right. Viruses can also mutate and change. Also known as pathogens, they will do whatever it takes in order to multiply in numbers and spread fast and quickly, and which requires a host such as ourselves. But our body has many ways of fighting these off, either with antibodies or with the help of vaccines. We'll be focusing specifically on the influenza vaccination, IV, or also known as the flu shot. Every year, the flu mutates and adapts to be able to resist against antibodies and previous vaccines, resulting in a stronger virus. It allows the virus to have more immunity against them. And because viruses such as the flu adapt and change over the years, new vaccines must be made each year in order to control and get rid of the flu virus.
So antibodies are Y-shaped proteins that are produced from the plasma cells in response to a specific antigen and are very important for the immune system. They bind with pathogens, which the body recognizes as alien, such as bacteria, viruses, and foreign substances in the blood and mark it for destruction. An antibody test is when they check someone's blood by looking at the antibodies, and if the antibodies are present, then it indicates that the person has had a past coronavirus infection. This is helpful because it tells the person if they have the antibodies to fight the virus the next time they get infected. So there are many different types of antibodies that protect the body against different kinds of viruses and other infections. The COVID-19 antibody are antibodies specific to the coronavirus and help the body fight against it. They take days or weeks to develop in the body after the body is exposed to the COVID-19 virus, but we don't know for how long they stay in the blood. Having antibodies to this virus are definitely important because it provides protection from getting infected with the virus again, but having them will not make someone completely immune to the virus because we don't know how much protection they provide. There are also scientists who are making COVID-19 antibodies from genetically engineered cows. A treatment available for treating hospitalized patients with COVID-19 called convalescent plasma therapy is where they use blood from people who've recovered from the coronavirus to give to others who have the coronavirus now, because the blood contains the antibodies to the virus so they can fight against COVID-19. However, antibodies from the cow are said to be more potent, safer, and longer lasting than the antibodies from convalescent plasma. Through genetic engineering, an artificial human chromosome is inserted into the cow so the cow would produce the human antibodies instead of the cow antibodies. Then they inject a non-infectious protein of COVID-19 into the cow so the animal can produce antibodies specific to the coronavirus. The scientists then collect the plasma from the cows 3 times a month, and the antibodies are removed from the cow plasma and are purified. SAB says that these antibodies are more effective than the antibodies in convalescent plasma because the cow's immune system is stronger than the human's immune system. Cow antibodies have more potential in treating the coronavirus, and scientists are continuing to develop more of these antibodies so we can be able to fight the virus. As you can see to the picture in the right, it shows how the antigen binds with the antibody at that binding site in order to mark it so the killer T cell is known to destroy those infected cells. Overall, antibodies are important because they fight off infections and provide protection against diseases, which is called immunity.
So like Purvi mentioned, antibodies can be produced by the body or by other organisms, like the cow example that she gave, but they can also be produced by pharmaceutical companies like Genentech, Moderna, Gladstone Institute, Vir, and more. And basically, these antibodies are used as a drug to stop viruses from infecting cells. And a little bit later on in the presentation, we'll go over how viruses actually infect cells.
Malaria, a Plasmodium infection. Malaria is a type of fever, both intermittent and remittent. Intermittent means that one is affected by the fever for a couple of hours, and then their temperature goes down for another couple of hours, and this pattern continues to repeat over the course of a couple of days. Remittent means that one's temperature is above normal for the entire day. It doesn't dip like the intermittent fevers. Malaria is caused by parasites that come from mosquitoes and insects that are carrying the disease. These parasites then travel to your liver and kind of just chill there for some time until they are big and matured enough to attack one's red blood cells, resulting in low blood sugar. They can also cause kidneys to fail and be the source of liquid accumulation in lungs. This results in various symptoms, including fevers, chills, headaches, nausea, and overall tiredness. Sadly, 210 million people get infected by malaria in a year, resulting in about 440,000 deaths a year. We do not have a cure yet, but we are working on it.
Ebola. Does anybody know what continent Ebola comes from? Write in the chat if you know which continent Ebola comes from. If you guessed Africa, you are correct. The virus originated in Africa. It is caused by an infection within a group of viruses within the genus Ebola virus, but only 4 of them can infect humans. Ebola virus, Sudan virus, Thai forest virus, Bundibugyo. Despite primarily infecting non-human primates, it's not spread by mosquitoes, water, or even air. It's spread through direct contact with infected blood, semen, feces, or other bodily fluids. Although the virus is thought to originate asymptomatically in the fruit bat, but it made an interspecies jump to the first non-human primates and then to humans. Symptoms or effects on the body caused by the virus include fever, headache, nausea, vomiting, and bloody diarrhea, chest pain and cough, stomach pain and severe weight loss, bruising, internal bleeding, and in later stages, bleeding from the eyes, rectum, ears, and nose. The death rate for an Ebola infection is anywhere from 25 to 90%, but it averages around 50%, especially with treatment.
So when you first see these two figures, it may be hard to find something that they have in common, but both of these figures are viruses, and even though they can be diverse, they have a lot in common in terms of structure. So viruses are not considered living because they cannot reproduce by themselves. Instead, they infect an organism with a cell and they take control of the cell so that it can reproduce the virus. The entire infectious virus particle is called a virion, and it consists of genetic material enclosed in a capsid, which is an outer shell of protein. And we talked about genetic material during our genetic engineering presentation, but it's basically something that gives you the characteristics that make up what you are. And an example would be a gene that determines hair color. So back to the virus, some viruses also have an external membrane envelope, which you see here. And so basically there's many different shapes that viruses come in. So this is the capsid, which is what is inside the envelope, but you can have an isohedral one, a filamentous one, and then a head-tail one. So for the head-tail one, I think it usually doesn't have an envelope because it can inject the virus into cells through the tail right here. So this is basically what goes inside of the envelope. So this is like showing that. So viruses are very diverse, kind of like we mentioned, there's like different shapes and structures, and then they also have different types of genomes and infect different types of hosts. So like some of them can infect different types of animals or organisms and stuff. So not all of the viruses that we know of infect humans. So kind of like you saw in the previous slide, there's the structure is very different, but you can see like there's the genetic material in here, and then also the protein envelope around it. Okay. So does anyone have any questions about like what we covered so far? You feel free to type in chat or like unmute if you want. Okay, if not, our next session is on December 13th from 10 a.m. to 10:40 a.m. And we'll basically be giving advice about creating a strong application and being confident when you're interviewing. So we'll be talking about like common questions that people will ask you during interviews or in an application, and then we'll also walk you through the steps to constructing a good response. So if you guys don't have any questions, we can go over some